All organisms need cell division to grow, mature, and maintain tissue. The process in which this happens is called the cell cycle. This cycle is split into four distinct parts known as the G1 phase, the S phase, the G2 phase, and the mitotic phase. G1, S, and G2 are all part of the interphase. In G1, the cell grows to nearly its full size and performs many of its functions that help the organism grow. Next comes the S phase, or the synthesis phase. It is an important stage because the DNA in the nucleus is replicated. The G2 phase is another growth stage where the cell finishes growing after the DNA has been replicated, and two centrosomes appear. When this is complete, the cell is ready to move to the mitotic phase. Mitosis is split up into four different parts. First comes prophase, followed by metaphase, then anaphase, and finally telophase. During prophase, we see the classic chromosome structure, which happens in a condensation process. At the same time, centrosomes appear and the nucleolus disappears. Metaphase comes next. In this stage, the nucleus disappears and the chromatids align. Microtubule strands then grow from the centrosomes and are attached to the kinetochore of each chromatid. Now the chromatids are ready to be separated. In anaphase, the sister chromatids are separated and are moved to the other ends of the cell. Telophase is the final stage of mitosis. The chromatids come together and a nuclear envelope forms. The chromosomes uncoil and return to an uncondensed state. Following mitosis is a process called cytokinesis. The cell is compressed by a contractile ring which divides the cell into nearly equal halves. The organelles are replicated and are divided between the two halves of the cell. This includes the rough ER, mitochondria, and the Golgi apparatus. The cell cycle can now start from the beginning. Cell division in our body is normally very precise, but sometimes a cell breaks free from its normal restraints and begins to follow its own pattern of cell division. This precancerous cell keeps dividing uncontrollably, and eventually a cancerous tumor develops. If there's a mutation in the genes that regulate cell division, a cell can become cancerous. The two genes that are involved are tumor suppressor genes and proto-oncogenes. Tumor suppressor genes prevent the cell cycle from happening, and proto-oncogenes encourages the cell cycle. If there's an imbalance between these two genes, the cell will divide uncontrollably. Some risk factors are smoking, secondhand smoking, family history, overexposure to UV light, radiation exposure, and much more.